Hey, good morning, everyone. Thank you, everyone, for coming. I'm Michelle Black. I'm the president of the Northwest Flow Cytometry Society. On behalf of the entire board, we'd like to welcome everyone here to the third annual flow cytometry meeting. It's wonderful to see so many people attending from across the Pacific Northwest and beyond today. I'd like to extend a special welcome and thank you and a thank you to our invited speakers, panelists, and those who have helped put together a fantastic program spanning cutting edge applications, high parameter panel design, data analysis, and careers in cytometry. We are also deeply grateful to our industry partners and sponsors, including our Lightning Talk and Happy Hour sponsors, for their generous support, which will help keep the meeting accessible and make today's program and networking opportunities possible. We hope you'll take full advantage of the talks, posters, vendors, conversations, informal discussions throughout the day, and that you leave feeling energized, better connected, and inspired in your own cytometry work. With that, we're going to get our morning session started. Couple things, just don't forget about our happy hour today. I know it's going to be a long day, but we do have the happy hour up the street at Pub 70 directly after the program. The board will be around. Garrett, Thane, and Alice are here from the board, and we are around if you have any questions or need help or any information you need from us. We're going to go ahead and get started with our first speaker. Our first speaker today is Daniel Chiu. He is a Bruce Montgomery Professor of Chemistry, Endowed Professor of Analytical Chemistry, Washington Research Foundation Professor, and Professor of Bioengineering at University of Washington. He is also a member of the Cancer Consortium at Fred Hutchinson Cancer Center. He obtained a BA in neurobiology and a BS in chemistry from the University of California at Berkeley in 1993, and then his PhD in chemistry at Stanford University in 1998. After completing his postdoctoral research at Harvard University, he started in the fall of 2000 as a assistant professor of chemistry at the University of Washington. He is the author of more than 200 publications and the inventor of over 200 issued patents. He is the recipient of various awards and honors, including the American Chemical Society National Friensis Award, the Pittcon Achievement Award. He was named a fellow at the American Association of Advancement of Science, the Keck Distinguished Young Scholar in Biomedical Research, and an Alfred P. Sloan Fellow. His research field of interest is nanomaterials, microfluidics, and new instrumentation for ultra-sensitive bioanalytical measurements and digital assays. With that, Daniel. Do you want to go in? Do you need a presenter mode? Thank you.
Thank you, Michelle, for the very kind introduction and for the opportunity to tell you some of the things that we're doing today. So my topic is digital flow cytometer, and I'll tell you what it is. But first off, a couple disclosures that are related to flow cytometry. The topic that I'm going to talk to you today is this instrument. We spun out a company called Panostics. We actually just passed regulatory on this instrument. So if you're interested, we're going to be working with Thane to deploy one at the South Lake Union Flow Core. So if you want to try some of those, just let Thane or me know. We also use some of the probes that we develop. We have a spun out company that work closely with Bio-Rad these are the P dots, or they brand it and they market it as a Starbright dyes. For the digital flow cytometry platform, a lot of people contributed, but primarily is Mengsha and Yuanhua that took the lead in a lot of the scientific technical development. The project and this instrument came out of an NIH ERCC consortium trying to develop methodologies, enhance rigor and reproducibility in the study of extracellular vesicles, extracellular RNAs. And so we got to work with a whole bunch of people within the consortium in the NIH. supported by the NIH grant. So what is digital flow cytometer? Uh, what it is a single molecule counter. So as the title shows, it's a high multiplex single molecule counter, and then it's in a high throughput flow cytometry format. So it's digital, parallel digital PCR. So a lot of the analytical measurement has been analog so far. Digital PCR is one of the first ones to turn it into a digital assay doing single molecule counting. And then after that, there were digital ELISA. So in this case, we are doing single molecule counting. And I'll tell you a little bit more about what the sensitivity and what the instrument can do. But very briefly, we have 4 laser lines that we shaped. They are like a light sheet that illuminates the entire cross-section of the channel. And then when the molecule or particle go through, then they would generate a photon spike. And what we get is a distribution of, these are single molecules, and these are the labels, say, for example, biological nanoparticles. So we validated the platform right now over 18 dyes that has, able to give you single dye molecule sensitivity. So these are absolute photon counts that we detected. And then as the molecule, it could be single dyes, it could be dye-tag single antibody molecules. When they go through, then they would have a large photon spike, which we detect. And the current instrument has 4 lasers at 405, 488, 561, 640, and also 12 single photon counting detectors. So you would have these kind of forest of photon spikes that come through in the different color channels. And the important thing is that for this platform, we can detect even the dimmest single molecule. So when you go down to single dye molecule detection, the photon emission is stochastic. So some dyes would emit more photons, some might bleach faster, and so on and so forth. And if you look at how much photon and the variability in the photons that a dye molecule might emit, that can vary by over an order of magnitude. And so when you do this kind of digital flow cytometry, you need to detect even the dimmest 1%, 0.1%. So we validated that for the dyes that we have tested, we can detect essentially 100%. And so the platform is based on a planar system. So one chip has 16 channels, 16 samples. You can run one sample, you can run 16. And if you put the 6 of them together, that will be a 96 format, also with the same pipette spacing and so on and so forth. The instrument is automated, so you just kind of put it into here, and then you plug a lure lock to control the flow, and then the system will run. And so for today, essentially, I think what we have is a single molecule counter in a high throughput, high plex fashion. So we try to develop different applications. So there are 2 applications I want to tell you about. The first one is studying single biological nanoparticle, and specifically, I'm going to focus on extracellular vesicles because this is what we initially set out to do. And then the second one, I think, hopefully, will be applying to more applicable to people that are doing high plex flow cytometry as well, and that is a single molecule assay that QCs antibodies. We didn't set out to do this, but then we see that there's a lot of variability when we do our single EV studies, and then we tracked it down to antibody variability. So we did develop an application package for that. So I'll tell you about that. So for extracellular vesicles, exosomes, there's a lot of interest. NIH has a 10-year program trying to study that. The first year, it was all excited about biomarkers and so on and so forth. And then the second half is mostly about rigor and reproducibility, because these particles are very heterogeneous. They're very interesting. There's a lot of promise, but then they're also very heterogeneous, very hard to study. There's a lot of biological nanoparticles in circulation. And so parsing everything out is not always the easiest thing. So we are involved in the rigor and reproducibility part and the methodology development part. So in my mind, if we really want to, if you look at the particle in circulation and blood, of course, there are the cells. I think most of you will study the cells with high plex and so on. But even more in terms of number-wise are the biological nanoparticles. EV is just one, but there are other non-membranous particles that are being reported, like exomeres. Was a Nature paper some years ago. Supermere collaborator reported. And these are all quite small. They are 25 nanometer, 30 nanometer, 35 nanometer. You have high-density lipoproteins that are down to maybe 10 nanometer, and they contain microRNAs. So there's a very heterogeneous populations of biological nanoparticles. We're looking at the other ones too, but for today, mostly just extracellular vesicles, exosomes that spans from, maybe 40 nanometer-ish or maybe a little bit smaller up to about 150. And they're generally characterized by the presence of tetraspanin. So those are markers that are classically defining what an exosome is. So we like to study them. And so the parallel is that if you look at how immunology, and I mean, you're more experts than me, developed is a really sort of synergistic, iterative cycle between methodology, flow cytometry, and the need of immunology. So I would say flow cytometry enable a lot of studies in immunology, and immunology go back informed what needs to be done in the flow cytometry, and that gone on for decades. And then now we're where we are with very powerful technologies and being able to do very deep and broad immunophenotyping. And so we think that we would need a similar tool to do that for biological nanoparticles. Currently, those particles are isolated by centrifugation or maybe a single marker immunoprecipitation and so on. So you're not going to be able to parse out the heterogeneity if you're using these kind of bulk methods. So the argument is that you really do first want to have combination of biomarkers that you can study rather than a single biomarkers like you do with cells. And this is a study from Weisslater's group using imaging that if you're doing 12 marker, you can trace the cell of origin from for the EVs. But if you do less than that, then you'll be hard to trace the cell of origin. So I think you do really need to have high plex ability to study it. And I'll show you that a lot of those biomarkers are present at very few copy numbers, 1, 2, 3. And so you do have, the requirement of very, very high single molecule sensitivity. And of course, when you do those, you need to have controls. If you do a multiplex, you singleplex and so on. So you really want to be able to have a high throughput format method. And also we're working on a sorting mechanism. So flow also allows you to do that. So just to give you an overview, this is a field that I think there's a lot of interest, but also there's a lot of technologies and so on. So at the level of single EVs, I would say that you have the nanoparticle tracking. We'll work with some of them. Those are really great screening tools, but that it doesn't have quite as much information content, very low plex and not quite as accurate. You can have super resolution imaging. So many of the data that we have, we cross-validate with super resolution imaging, very, very high sensitivity, a lot of information content, but the throughput and multiplexing is limited. Of course, there's the flow cytometers. Most of those are for cells and EVs, but there's a couple that came up that has more, tailored for EV. So I think we're here to talk about Cytoflex new nano, Cytoflex Nano, and then the Nano FCM and so on. So we'll compare to some of those. So if you're interested, we can talk more about it. So for us, we're thinking that, we really want to be able to offer a method that is high plex, high throughput, so you can really do deep immunophenotyping on single EVs. So when we do this in comparison, we do see that a lot of the particles, if you're using the existing commercial instruments, they're designed for cells, but then trying to make it more sensitive for EVs that you miss quite a bit. And so we did this study as published in Journal of Extracellular Vesicles with a bunch of other people within the consortium. And so in the top-down flow cytometer, they're designed for cells. You're probably good to maybe 180, depending on how you look at it. The Cytoflex Nano is very, very sensitive for scattering, but if you look fluorescence, it's still quite limited. And so generally, I would say that there's two things. First of all, those two methods are very complementary. If you're looking at particle larger than 100 nanometer-ish or 200, the existing conventional flow, I think, is really great. But if you want to go to very low and small particles, then you probably want to have a single molecule sensitive method. And so this is sort of using a reference sample that we got from Bob Coffey's group, part of the consortium. So one of the things when we did the study too is that, well, because the two techniques are very complementary, then, like to use both, right? You can go to the larger particles, and then the throughput for the conventional would be great for the large particles. You can see all the tiny particles using our method. So they're very complementary. And so one thing we would like to do is that, well, if we use beat calibration, we can define the limit of detection, the MESF, and so on and so forth, so we know which population that we are mostly looking at using the different techniques. So one thing with the beat calibration when we studied it is actually, especially when you go down to single EV nanoparticle level, is that the beat calibrations are very high precision measurements, great for reproducibility, but it's actually very low accuracy measurements, meaning that it doesn't really tell you the absolute, LOD. If it's 10 p MESF or 20 p MESF, the actual MESF is actually can be quite variable. The study we did was that if in order for the two data sets to be concordant, we can say, well, we have to toss out our data so that they're concordant. This is our threshold analysis to show what is the LOD, and then that LOD is different than the bead calibration. So one of the things going forward we're hoping to do is to calibrate the beads using this kind of a single molecule bottom-up approach, so that we can make these kind of two sets of approaches that are more complementary. So with the method, I'll show you some of the data that you can get out from that. So of course, you can do high-plex. We can do 5-marker, 7-color with no compensation at all. And then with compensation, you can go higher, and then you can get very high-plex immunophenotyping. For every subtype, you can get an absolute concentration measurements out. So I'll tell you how to do that. And also, these are digital single molecule counting, so we can count the absolute copy numbers on each, of each marker on the EB. And these are some examples. So for the absolute concentration, what we do is that we track the velocity of every particle or molecule that come through. We know the cross-section of our channel because these are a planar system that we fabricated. And so if you sample enough particle that go through, then the velocity you can translate into a volumetric flow rate. So if you have enough sampling, 100 particles, molecules, or 500, even though you have, I don't know, a nanoliter or 5 nanoliter that have gone through, we can tell you pretty precisely exactly how many nanoliter down to the single digits. And since we count every single molecule as long as it's got a dye tag to it, then we can divide the two and get absolute concentrations out, including single antibodies, which I'll come back to because that turns out to be really helpful QC for antibodies. We did that with EVs, obviously, and when we did that, we also developed a protocol where we dry all the EVs and still being able to image them and without aggregation and bleaching, so we can correlate the two methods and cross-validate, and they cross-validate pretty well. So that's a concentration, absolute concentration measurement. We can also do absolute copy number determination. So what we do is that every single run we have, there's going to be some free antibodies. So every run is internally calibrated with the single antibody signal. We also have our labeled EV or biological nanoparticle or whatever else we have. And so when we have those two sets, we can do a deconvolution and then get the absolute copy number on the EVs, in terms of the biomarkers. So these are some of the studies, systematic study we did. You can get, if you have, 70% one copy and 30% two, you can back that out pretty well. Same thing here. And I'll also show you the application of this, not just for EVs, but again, for antibody QCs that we're really hoping that people will take a little bit more time to do. So I'll describe that, but using the same capability where we would tell you what is the number of fluorophores per antibody, number of side dyes per PE tandem, things of that sort. So all of those are digital single molecule counting measurements. They're absolute measurements. We do one analog measurement, and that is we use the intensity of a membrane dye to correlate with the size of a membrane particle, EV, and so on and so forth. We opted for the system is all fluorescence. We opted for all fluorescence and not do scattering. Scattering, of course, is a very useful signal, but when you go down to biological nanoparticles, the refractive index actually may be different from particle to particle. You don't have a really linear curve in terms of scattering intensity and size. So they all have their own pros and cons, but we opted for fluorescence. So these are some of the things that we thought about with scattering. But fluorescence, you also have to be careful in terms of the dye behavior. One of the things people haven't thought as much about is that you also, because these are very small particles, you also need to have a sufficient number of membrane dyes per particle. We have screen ones that are really great, but then we got like less than 10 dyes per particle. And then in that case, your intensity is dominated by the Poisson distribution rather than the size variation. And so we have a lot of different metrics to make sure that that's the right assay. So we have developed them in the protocol. So if you're interested, we can talk more about it. Again, this is a single molecule fluorescence counters, and there's a lot of assays out there that are fluorescence based, including for EVs. So what I've shown you before are just simply immunophenotyping and things of that sort. But you can access the internal cargoes if you're interested. We have membrane permeant dyes that gone in and label RNAs or DNAs. We can have molecular beacon that go in and recognize specific sequences and so on and so forth. So all of those you can do, and those just involve new assay development. And we'll continue to try to develop new assay development to expand the capability of the platform. So overall, for EVs and biological nanoparticle, we think that is a new capability that allow you to detect every fluorescent molecules. You can immunophenotype pretty high dimensions, at least for as far as EVs concerned. For cells, you go much higher plex, but for EVs, is really limited at the single molecule level. Is really limited by the dyes, and also compensation gets very tricky when you're in a very very photon starved regime. You can do absolute concentration measurements and absolute copy number distributions. I mostly just tell you what the system can do for EVs, but we're also working on various assays for lipid nanoparticle, viral vectors, AAVs. AAVs are 25 nanometer. So if you look at the absolute copy numbers on the particle, you have a permeant cyto dye, you can actually one shot within a minute be able to see which ones are empty, which ones are intact because of the copy number, and so on. So that's, I think, the part that I just want to give you an overview of EV analysis. This is what we set out to do. But for the remainder of the talk, I actually want to tell you some of the things that we did by accident. Didn't intend to do that in the beginning, but turned out to be very useful for us, which I hope will be useful for you too. And that is it's a single molecule assay trying to do antibody QC. So for EVs, when we did our work, we do see variabilities in results. In the very beginning, we thought it was our platform, it's a new platform we're building, is it the alignment and so on. We spent like months on that. Then maybe it's the device, maybe the fab has some variability, and then maybe it's a sample, and then we do split samples, and it turns out that there's a lot of antibody variability. And that is even more sensitive if you're doing EV type of analysis than if you're doing cell type of analysis, because there's so few antigens on the EV. So, the antibody reproducibility, I think, is a big, in people's mind, on NIH mind 10 years ago, we all have to put in authentication in our proposal, I think in large part because of worries about irreproducibility. And if you're doing quantitative flow cytometry, then, some of these things are even more important. So for example, if you want to do absolute, marker expression level, using a calibration bead, you will get a bead intensity, PE/MESF equivalent, and then you rely on maybe 1 PE per antibody to then, calculate how many expressed antigen that you have. So there's a number of assumptions that are baked in. Concentration is important. KD is important. So all of those parameters are quite important. And so is even more important when we do EV analysis. Again, I've shown you some data, some EV has only 1 copies of an antigen or 2 copies of an antigen. So if these factors are not really well controlled, you might be generating artificial subpopulations because it's not binding because of the antibody and so on. So a lot of it is to do with the quality of the antibody. And so there are 3 sets of things that we do, and they're pretty routine that we do now, and they're pretty easy to do. The first one is just to get an absolute concentration now. I show you that we can detect every single molecule and then get the volumetric flow rate very accurately. So all you have to do is to pipette as little as you can, maybe 1 microliter, as long as your pipette can handle it, doesn't evaporate, and then we'll dilute it maybe 100 to 1,000 times. You pop it in the instrument, and then within a minute, you can get your absolute concentrations out. And so, and if you do that, you do see variabilities, from dye to dye, and also from time to time and from lot to lot. And also, if you have antibodies, some of the antibodies tend to maybe aggregate over time, and you can see that too. And also for some vendors, and we've got antibodies from different vendors, even a qualitative run in the very beginning, you see that, well, this one has more aggregate than the other ones. But over time, you can also see that from when we do this intensity analysis and so on and so forth, you can see that even though they're very small aggregate, they're very difficult to detect with conventional bulk methods, you can see it in the system pretty well. In this case, we saw there's sometimes less than 1% of aggregate, and the aggregate can be quite small, maybe 50, 60 antibodies, but that would affect your result in terms of the labeling concentration and also the non-specific bindings, since aggregate can do that. So that's one assay I would say we do pretty often now, just do a concentration measurement. If we haven't used the antibody for a while, especially, and then we just drop it in and make sure that the concentration is right, especially for new assays when we need to know the concentration in order to run a good titration, make sure that we are running it at saturation labeling. The other thing is FP ratio. If you're trying to do quantitative flow cytometry, this is an example for they are all PE conjugates, so you can see that they can be quite variable. You can have antibody that are labeled with the same dye. You can have different degrees of labeling or FP ratios. And incidentally, if your dyes have more dyes on an antibody, that would also change the KD, and then that would also change your optimal labeling concentration. So those are just examples of the fraction of 1s and 2s and 3s for PE. PE generally is thought to be a pretty big molecule, so it's 1 to 1, that makes sense, but in actuality, it can be variable. And then for this case, for Alexa 647 labeled antibodies and clone, you can have, in this case, there are 3, 1, 2, and 3 equal portions. In this case, there are a little bit more. You can also get PE tandem. So in this case, we look at the intensity of single Cy5, and then you look at the conjugates, and we have 2 different vendors, and you see the conjugation ratio is different. When we look at a single antibody brightness, one is actually 50% brighter than the other ones, and these you can also potentially get lot-to-lot differences. Finally, the other thing we do is KD measurements. So there are different ways to measure KDs. If it's fluorescent antibody that you use in flow, that's actually pretty easy, because in this method, we're seeing both the bound population and also the free population. KD is free over bound, right? So you can get really accurate KD measurements, subtle changes you can see. But also, if you do competitive assays, you can also get KD measurements from non-fluorescent antibodies and so on and so forth. So overall, we're hoping that this will be something that we're doing that all the time now, that people adopt it, because it's pretty easy to do. Again, you just need to have something you can pipette 1 microliter, and then you have to actually dilute it about 100 to 1,000 times, depending on the initial concentration. One chip can run 16 samples, so you just pipette it into one reservoir, and then per sample, maybe a minute, you'll get all the data out. You'll get your all your quality control. If you're spending a lot of time on your cells, on your panels, it seems like it might be a good thing to do, just to make sure that the antibodies are behaving properly. So if you're interested in doing that, I think most of us are local. We're working with Thane to deploy it at South Lake Union. So if you say, hey, I want to check my antibodies, maybe I can just see what the results look like, then just let us know. Be really excited to have you take a look and try, and to see if it's useful and is convenient for your workflow or not. So with that, I think that's all I got. Thank you very much. I think I'm supposed to come down or answer questions. Yeah, as long as they're fluorescent, we can do any kind of binding stuff. We're also developing assays for sort of therapeutic antibodies too. These are not fluorescent. You can just do a competitive assay. You can get it out because you can, unlike SPR and all of those, these are free solutions. You can see both population. If your antibody is not fluorescent, a drug, for example, you just need to do a competitive assay using a fluorescent antibody, and then you get the results out. So we can see antibodies, but if you're saying that they are individual antibodies inside a cell, that will be difficult for us for two reasons. One is that um, these are biological nanoparticles, and so for cells, one molecule in a cell is different than one molecule in a nanoparticle just because you have a lot more volume, so the background will be higher. And second is that this platform is designed for things that are less than 2 micron. So we're trying to do nanoparticle. Yeah, I think some of the conventional flow, because they can do both, while it's flexible, it gets a little bit tricky when you're trying to go down really small. I think quantum dot is useful, but I don't think that they're very widely used as some of the other dyes. They're out there. BD has the real dyes that's got good spectral. We developed the star brights. They're very bright. So when we do single particle, comparable size water soluble quantum dot and the P dot, the star bright, they're about an order of magnitude brighter. Quantum dots, you cannot do excitation multiplexing so easily because you don't have absorption feature. But you can do emission multiplexing. So we did a lot of comparison between P dots and quantum dot because they're both kind of nanoparticle. And of course, there's a lot of different considerations, but I think it depends on what you're after, I suppose. For flow, the lifetime is also a little bit long for quantum dots, usually the 20 nanoseconds, and you want to have shorter lifetime since you're such a short integration time. So we can talk more about it. Yeah, sure.
I'm going to introduce the next speaker. She's Lisa Dornisch. Lisa is a research associate on the experimental immunology team at the Allen Institute for Immunology, where she studies the role of B cells in health and disease. Her work leverages high-dimensional spectral flow cytometry, ex vivo immune cell cultures and assays, and immunofluorescence imaging of tissue and primary cells. Lisa earned her BS in biochemistry from the University of Puget Sound, and prior to joining the institute, she supported clinical and preclinical research in maternal and fetal medicine at Medigan Army Medical Center in Tacoma.
Good morning, everyone. Thank you for having me here to speak today. Again, I'm Lisa from the Allen Institute of Immunology, and today I'm going to be talking about how we use spectral flow cytometry to study B cells. Great. So B cells are a critical component of the adaptive immune system, owing to their ability to mount durable and specific responses to a range of different pathogens. When a person gets exposed to different pathogens such as virus or bacteria, a B cell can encounter its cognate antigen through surveillance in a tissue such as a draining lymph node. A B cell receptor will bind its cognate antigen and become activated and start undergoing proliferation, and with the help of T cells can enter into a germinal center. The result of the germinal center will be the differentiation of memory B cells, which will recognize the pathogen in subsequent exposures, as well as plasma cells, that can secrete antibodies to neutralize the pathogen. All of these processes are extremely important for B cells to keep us healthy to all the different pathogens that we can encounter. Although B cell mediated responses aren't the only way that B cells help keep us healthy. B cells serve many different functional roles, including antigen presentation to cells such as T cells. They are involved in co-stimulation, as well as tertiary lymphoid organ formation, which can concentrate an adaptive immune response to a site of inflammation, which is important in contexts such as cancer. And they can also act as immunoregulatory molecules by secreting cytokines that can either stimulate or inhibit different immune processes. So given all the functions that these B cells can serve, it's important to keep this into account when looking at the different phenotypes that we're looking to study in the context of health and disease. And many groups have looked to do this, including the Bendel Lab at Stanford, where they have done a surface screen of over 300 different molecules looking at human B cells across 4 different tissues. They looked at lymph node, kidney, bone marrow, and peripheral blood, and were able to come up with distinct B cell phenotypes through high dimensional analysis that could correlate their phenotypic identity to the function that they serve. And for the rest of the talk today, I'm going to talk specifically about the peripheral B cells in blood. And looking along the axis here, we have our B cell maturation as well as their abundance. We have the different isotypes that B cells have and a couple different populations that I wanted to highlight. Starting with our transitional and immature B cells, these have recently emerged from hematopoiesis. They express the unswitched isotypes of IgM and IgD, and they have they're in circulation and they're not antigen inexperienced. We have our naive cells, which are abundant in circulation, that also express IgM and D, and they can co-express IgM and D at the same time, and again are still antigen inexperienced. Moving on into the differentiation of B cells, once they experience antigen, we have different memory B cell markers that we can look for, specifically CD45RB, the clone MEM55 is the glycosylated form of RB and is an early memory marker for B cells, as well as CD27 as another memory marker that we can look at. And this is where we'll start to see our class-switched isotypes, although we have seen unswitched memory cells as well. We have other atypical tissue-resident and effectory B cells that we can study as well. And we have our plasma cells, which can be identified by CD38 and CD20 negative. Our group is currently working on looking at the heterogeneity of these plasma cells, so keep an eye out for a publication upcoming on that. We also have different memory populations, such as CD95, which we hypothesize to be a recent immigrant from a germinal center reaction. And we also have this very interesting CD19 high CD11C effectory memory B cell population that I'm going to talk a bit more about today. So this population has mixed expression of CD27, and we know it as CD19 and 20 high and CD11C positive. And while it has a unique transcription lineage, it is known under many different names in current research. So you might have heard of it as atypical B cells, age-associated B cells, double negative B cells, and much like the puma conclure, where this is one of the most widely distributed mammals in the Western Hemisphere, it has been known under all of these different names, despite talking about the same feline. I grew up in San Diego, knew it as a mountain lion. People in Washington know it as cougar or panthers. But what's important to realize here is that all of these different publications are talking about the same cell, which we'll refer to as the effector memory B cells. And this is an interesting population because we've seen it expanded in different autoimmunities such as lupus. We've also seen it involved in malaria infections, but it's also been shown to undergo normal B cell maturation processes such as class switching and differentiation into antibody secreting cells in response to flu or COVID vaccine. So there's kind of an opportunity here for the field that needs to kind of limit the confusion of all these different names and focus on one name so that we can really understand the role of the specific effector B cell, because it's seen to have numerous implications in both health and disease. So what I'm really going to focus on in my talk today is the characterization of B cells within spectral flow cytometry and how we study them at the Allen Institute for Immunology. And first, I want to talk about our B cell flow cytometry processing. Given that B cells are they're not a very abundant population, we've developed our protocol to kind of improve the viability and cell retention at every step. And this starts all the way at the thaw of PBMCs. And we supplement our thaw media with 20% FBS. B cells really need the extra fetal bovine serum to keep them happy throughout our processes, and they can be sensitive to any protein degradation in the FBS, so it's important to thaw it at 4 degrees. And we also, given that our B cells are not very abundant, they're only 5 to 10% of PBMCs, meaning that if you want 1 million B cells, you need to thaw about 20 million PBMCs. And given that, we often do a B cell enrichment using a negative selection kit where we can leave our cells of interest untouched and avoid any unwanted activation by binding the cells. And so with our negative enrichment kit, we can get our enriched B cell population, and we want to make sure to immediately use those cells because as soon as they start to sit by themselves, they tend to become very unhappy and will die off. And usually the next processing we have for that is flow cytometry, where we seed cells at about 500,000 to 1 million cells per well. And B cells have many FC receptors, so it's really important to FC block them. And we always viability stain to make sure that any results we're interpreting are from our live populations. We can also do intercellular staining on these cells. We'll want to make sure to use the right fixatives for the different epitopes that we're looking at because the fixatives can affect them. And any incubation we do, we keep at 4 degrees or on ice to, again, retain the cell number and viability. Within our data acquisition for our single stain controls, we use B cells whenever possible, and we'll do lot-matched controls between experiments. If we don't have the cell numbers, we'll use beads and make sure the spectral signature is very clean for our mixing. For our on study, we usually collect 500,000 to 1 million events. And for our QC development, usually 250,000 is enough to look at our different memory populations that we're looking to get at. From there, we do manual gating of the different B cell subsets that I previously described in either FlowJo or Cell Engine, and we complete a lot of our analyses in R after that. So going into the panel that we've developed, here we have our B cell identity markers. We use both CD19 and CD20. We have our different isotypes, including our class-switched IgG isotype that we can look at. Then our memory subsets. Again, it's very important that we use the clone MEM55 to get the glycosylated form for our early memory marker. And we also use CD27 for our most common memory populations. And we have our CD11C to look at this effectory B cell population that I mentioned earlier, as well as different markers, CD38 and CD24, that we can look at our naive and transitional B cells. Once we've developed our panel, we can use technologies such as Cytec Spectrum Viewer to look at the spectral signature of all the different colors within our panel and to look at our similarity matrix. We always want to keep in mind any co-expressed markers in our antigenic density when developing these panels. And once we're confident, we can move forward into testing this on samples. So this experiment was from 1 million events of PBMCs collected. It was gated for lymphocytes and single cells. And then here I'm showing the live cell gate, which will go through our different B cell phenotypic populations. So we have our lineage channel that includes markers for different cells, different markers we don't expect to see on T cells, such as CD3 or CD14. And we can do our first pass gate of B cells looking for CD19 expression. We can then gate by CD19 and CD20, where the majority of our B cells will fall into the double positive, and our plasma cells will be CD20 negative, so we can look at those there. From our B cell population, we can look at our effector B cells, where you can see the CD19 high CD11C population, as well as its mixed 27 expression. Again, this is those effector B cells that I mentioned earlier. Going back to our not effector B cell gate, we can look at different memory populations, including our CD95 memory B cells, as well as our main memory gate, CD27 by CD45RB, where we can look at our early core and late memory populations. From the double negative, we can look at our naive and transitional B cell gates. And this is our basic panel that we've used to apply to different cohorts that we have at the Allen Institute for Immunology. And the first example I'll be talking through is a study where we looked at the B cell response to flu vaccine in healthy adults. In this cohort, we had young and older adults who received a flu vaccine in the 2020 to 2021 season. We have 3 different time points where we collected samples, the first being their day 0 or baseline, and we have day 7 where we can look at their acute response to the vaccine, and day 90 where we can look at their durable responses. We collected PBMCs from these different time points and then looked at single-cell RNA sequencing and spectral flow. I'm only going to highlight a very small aspect of the paper, but I have it linked down here and at the end for your further review. And thinking back about our effector memory B cell population within the single cell RNA sequencing data, we found that both young and older adults had a robust response to the vaccine. Looking at their day 0 to day 7, we saw that the CD27 negative effector population increased in both of our groups. Whereas when we looked at the class-switched isotypes of the CD27 negative effector B cells, we saw that the younger adults had a higher frequency of class switching compared to older adults, which we can also see in this representative flow cytometry plot looking at all B cells by IgG. At day 7, between younger and older adults, we see a reduced frequency of the IgG. And this result is an important finding because we know that class switching to IgG is an important process in an effective response to the flu vaccine. So this data would suggest to us that the older adults are not developing as a robust immune response when given a vaccine such as the flu. Another study that we were able to apply our B cell phenotyping panel to was looking at B cells in the rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease that's characterized by painful inflammation in the joint. And this is actually a progressive disease where people can be diagnosed as at risk for rheumatoid arthritis based on different factors detectable in their blood, such as anthracyclinated protein antibodies, or ACPA as I'll refer to them, as well as rheumatoid factor. They can find these patients at risk, and while some, but not all, will convert to clinical RA, which is diagnosed by the inflammation and pain in the joint. So our group is interested in kind of studying this progression to rheumatoid arthritis and understanding the cellular and molecular states that might be leading to someone in an at-risk state converting to clinical and rheumatoid arthritis. And we have a longitudinal cohort where we have individuals that are identified at risk, as well as individuals that have clinical arthritis. And we also have those in our cohort that were identified as at risk that have actually converted to clinical RA during our study. And looking at the PBMCs from those patients, we ran a pipeline spectral flow cytometry panel, which I won't show that today, but I will show what we found in those results was that leading to their progression to diagnosis of rheumatoid arthritis, which is where you'll have that inflammation and pain in the joint, we saw this expansion of naive B cells prior to their actual diagnosis. And so we wanted to adapt our B cell panel to look at the functionality of these naive B cells before progression to rheumatoid arthritis. And here we have our B cell panel that we've adapted to include our cytokines. If you remember from the beginning of the talk, we know B cells can produce and secrete pro or anti-inflammatory cytokines that can mediate different immune response processes. They're also known that they can produce rank ligand, which is thought to be a link to pathogenesis in rheumatoid arthritis. And what we did here was we took healthy individuals and individuals at risk for rheumatoid arthritis, where they were identified here as being ACPA positive. And we have PBMCs from those donors that we isolated and stimulated for 48 hours using TLR9 ligand and CD40. Within the final 6 hours, we gave them PMA and iamycin, which increases their cytokine production. And we looked at these by intracellular flow cytometry. And interestingly, what we found was looking within those naive B cells, since we saw expansion of them as they led to progression towards rheumatoid arthritis. We found that the individuals at risk, when stimulated, produced higher amounts of rank ligand, IL-6, and TNF. And it's this very interesting result because we're seeing this skewing towards producing pro-inflammatory cytokines, even though there's no inflammation in the joint at this point of these patients. So those are two examples of how we've used this panel to look at the role of B cells in health and disease at the Allen Institute, and how spectral flow cytometry is a powerful tool in identifying driver B cell phenotypes in human tissues. We've talked about how working with B cells requires special consideration for sample processing, all focused on retaining higher cell numbers and viability, and kind of walked you through our different B cell identities that we can detect in the peripheral blood. We've looked at their surface phenotypes and cellular functions and outlined the sample antibody panels and gating approaches that we've used, as well as highlighting how we've employed this panel to look at different cohorts, specifically looking at healthy adults and their responses to vaccines, as well as looking at patients in subclinical rheumatoid arthritis. So with that, I'd like to conclude with a quote from this review from Sister and Allen that I feel gives a nice perspective about the current state of the B cell research field. With so much already known, B cell biology might be considered done, with only incremental advances still to be made, but instead there's great activity in the field today with numerous major challenges that remain. For example, efforts are underway to develop vaccines that induce broadly neutralizing antibody responses, to understand how autoantigen and allergen reactive antibodies arise, and to harness B cell therapies to correct autoantibody mediated diseases, making it evident that there is still an enormous amount of that we do not know about B cells, and there is much work to be done. And with that, I'd like to thank the Allen Institute for Immunology, specifically Marla Glass, who's led the B cell research team there, and our different project teams for immune health and aging, as well as our rheumatoid arthritis project team. And a huge thanks to our flow cytometry team, including Vaish, who's put a lot of work into helping us develop these panels. And with our lab operations team, none of this work would be possible. We'd also like to thank our data apps and software development team, as well as leadership, and all of our collaborators, including UCSD, Colorado University, BRI, and Fred Hutch, along with the Paul Allen Family Foundation and the NIH, and of course the study participants who none of this work would be possible without. And with a big thank you to the whole Institute team for all of their support and contributions to this work. I wanted to leave you all with some resources. All of this data that I went through today, along with much more, is available on our website, where you can look at the flow data as well as other interesting data sets of flow cytometry for our immune health across aging, including the papers that I discussed today, as well as our progression to RA. And with that, I'd like to answer any questions. Yes.
Such a rare peripheral population, like I think it was C.7 positive, or that count is that low. I was wondering how your team approaches verifying that state is real.
Yeah, so there's a lot of QC work that I wasn't able to show today, but we really try to focus on having not only technical controls, but also biological controls. So populations we know that'll express, for example, CD27 or our markers of interest. So we'll try to validate both technically with any kind of FMOs, as well as run higher numbers that we can be confident that the population will replicate between donors and between experiments, but kind of utilizing biological positive controls as well. Sorry, lights. Yeah, we have looking at the transcriptional lineage of the population, we've defined it as Tibet positive and ZEB2 positive. And I think the point of the atypical B cells is that we've actually seen specifically with this response of with this population looking at the CD19 high, CD11C positive, I think there's still a lot that's unknown about this population, and it might have other markers such as CXCR5 that you're showing. And our interest is specifically looking at how the role of these cells is involved in both immune response in healthy contexts such as vaccine, as well as their different roles in different autoimmune diseases. So I think that's a great point, and that there's probably a lot to be learned more about that population and probably more populations that we've yet to discover. Thank you.
The next speaker is Katharine Schwedhelm, and she is a researcher manager for the HIV Vaccine Trials Network, HVTN, Endpoints Laboratory at Fred Hutchinson Cancer Center in Seattle. She gained experience in flow cytometry and instrumentation at Benaroya Research Institute before moving to Fred Hutch, where her focus has been directed towards high parameter panel development to support HIV and COVID-19 vaccine studies, and cytometer standardization and characterization. Katharine also supports the Cape Town HVTN laboratory, facilitating panel transfers and ensuring cross-lab assay and instrument concordance, and has been an instructor at the African Flow Cytometry Workshop.
Thank you very much for the introduction. I am going to be speaking today about high parameter panel development, flow cytometry characterization and standardization, and assay validation for vaccine clinical trial testing. So the topics that I'm going to cover today, just a brief overview on the HVTN and the intracellular cytokine staining assay that we employ, our approach to flow cytometer characterization and standardization, some principles of high parameter panel development, and then finally finish with how we validated our ICS assay for clinical trials. So the HIV Vaccine Trials Network was established by the NIH in 2000. It's a large domestic and international network of both clinical sites and processing laboratories. There's centralized immune monitoring laboratories as part of the HVTN. It started off with 2 labs, one in one in Seattle and one at Duke University. And then in 2012, we added 2 more labs, one in Cape Town and another in Johannesburg in South Africa. And the Seattle and Cape Town labs use the intracellular cytokine staining assay to monitor T-cell immune responses to vaccine candidates. And our labs are recognized for our expertise and approach to quality control and validation. We use the intracellular cytokine staining assay to monitor T-cell responses to vaccine candidates. This assay takes place over 3 days. On the first day, we thaw cryopreserved PBMC and then rest them overnight to recover their function. Then on the second day, we stimulate the cells with peptide pools or other antigens of interest for 6 hours or more in the presence of Golgi block. And then at the end of the stimulation period, we add EDTA and we store them overnight in the fridge. And then on the third day, we perform viability staining followed by surface staining. Then we lyse, fix, and permeabilize the cells. And then we perform intracellular staining. The samples are acquired on the flow cytometer in high throughput, and then the resulting data are analyzed in FlowJo. So now I'm going to move on to discuss how we characterize and standardize the cytometers that we use to acquire these samples. The procedure that we use was developed by our colleagues at the Vaccine Research Center. And while we do have BD instruments, this can be used for instruments from other vendors. As when we bring a new cytometer into our lab, it undergoes a very thorough characterization and optimization process, followed by standardization and validation. And then for all of the cytometers that we use, we perform ongoing quality assurance. And then just in the interest of time, I'm only going to speak about voltage scan to determine optimal operating voltages, how we establish the QC targets, and then daily QC prior to use. But afterwards, I can talk about the other aspects if anybody has interest. So the voltage titration or voltration is performed to identify the optimal voltage for each detector. There's two procedures that you can use. One of them involves stained cells. You require 1 fluorochrome per detector for a conventional instrument, or enough fluorochromes to cover all of the detectors if you have a spectral. The other procedure can use unstained cells plus a mid-range hard dyed bead combined together. Both of these procedures involve incrementing the voltage of each detector and recording data at each increment. The stained cells procedure, you have to do this 1 detector at a time, so it's slower, but you get a measure of the true stain index, whereas the unstained cells plus bead is much faster because the bead fluoresces in every single detector, so you can increment each detector or all the detectors at once, but you only get a relative stain index measurement. Then once you've done all your voltage increments, for each voltage increment for each detector, then you calculate the stain index and you plot it against the voltage to obtain a curve, which I'm showing in blue. Not sure if the pointer is working. No, not working. Okay. Stain index curve is in blue. And then also you can extract the standard deviation and then the MFI of the negative population and also plot that on the same graph. And those are the lines in green and red on the plot to the left. Additionally, you can concatenate each FCS file from each increment and show it all on one plot. And then together, these are going to help you determine the optimal voltage for the detector. And this is going to be where the stain index is maximum or close to maximum and where the spread of the negative is low. So on the plot on the left, it's the vertical, showing the vertical line at a voltage of 500. And then on the plot on the image on the right, these are concatenated cells and the optimal voltage is circled in the red box. So once you determine the optimal voltage for each detector, then you can set target medians for your QC particles, and this allows you to standardize your cytometer on a daily basis back to those optimal settings. You will set all of your detectors to their optimal voltages and then run a mixture of hard dyed beads. We use 6-peak and supra. And then you're going to obtain the MFI of the positive peaks for each bead type. Then you choose the optimal bead for each detector, and this is going to be the one that fluoresces or has the highest intensity without exceeding the linear range of the detector. And then that associated bead MFI becomes your daily target moving forward because it ties back to the optimal setting of your detector. So what this looks like is an example for the red laser detectors off a conventional instrument. We set them to the optimal voltages. We ran the mixture of 6-peak and super beads, and then I selected the optimal bead for each detector, and those are the ones that are in the red boxes. So 6-peak for the R660 and super for the other two. And then the bead target is just the bead MFI rounded to the nearest 1,000. And then on a daily basis, before running any samples, we'll put on our mixture of 6 and super beads. And for each detector, we're going to adjust the voltage as needed so that the MFI of the bead today matches our target. And that's what I'm showing in the bottom. So when you have standardized instruments, there's many benefits to this. If you have multiple instruments, you can standardize them to each other and then use them interchangeably. You can also establish standardized gating templates. And this is really important because it reduces the bias in gate placement. And this works because when you have standardized instruments, because the median fluorescence intensity of expression of a marker is similar between participants, that positive population is going to always fall in the same place. It also can help you differentiate between instrument issues that you might have, like if you're looking at your data and something looks weird, it could be an undetected clog, or technical issues such as missing stimulations or incorrect antibody titers, because you know that your cytometer at its base is operating optimally. And I'm going to talk briefly on best practices for high panel parameter design. This topic really does warrant a whole lecture on itself, and there's not really time for that, but just some best practices. So at the very foundation of designing a successful high parameter panel is optimizing and standardizing your instrument, and also really understanding your instrument, its configuration. What lasers do you have? What detectors do you have? It's no good trying to use a brilliant UV fluorophore if you don't have a UV laser. Also really useful is your instrument spillover and spreading matrix. Then when you're starting to put your panel together, you want to identify what markers are critical. And then you also want to understand your marker expression and then co-expression of your markers. And it also helps to develop a basic gating strategy. Then when you're starting to assign fluorophores to these markers, you want to consider factors that can influence the performance of your panel. I'm sure most people have heard this, assign bright markers to dim fluorophores and fluorochromes and vice versa. But also you want to think about how your spillover or spectral overlap of your fluorochromes are, and you want to assign them to markers that are not co-expressed, and that goes back to your marker selection. And then also using your instrument-specific spillover and spreading matrix, because that's going to tell you which detectors have overlap. There are a lot of resources out there, in particular, the Optimized Multicolor Immunofluorescence Panels publications, or OMIPs. These are publications on optimized flow panels. You can start, this is a good place to start, because potentially somebody has created a similar panel to the one that you are trying to design, and you can use it as is, or you could take a panel that somebody has designed and use it as a backbone for the panel that you want to develop. There's lots of great in silico tools to provide feedback. All the main vendors have one. These assist in panel design. They give you similarity indexes and can give you an idea of whether your panel will perform well before you even test it out. Then once you have a panel that you want to move forward with and you order all the antibodies, very, very important to titrate those antibodies before you test them in a panel. Important to realize the best titer is not always the one with the highest stain index. You want to be able to balance, being able to discern your positive and negative populations, but also reducing as much as possible the spillover in your panel. Then once you've run that panel, you want to do a thorough assessment of the panel performance. A great way to do that is using multi-graph overlays to check for hidden spreading or n-by-n matrices. In our lab, we only actually run the FMOs once we've kind of identified a final panel that we want to move forward with. They're a lot of work, so it's really only at the end where you want to implement this. And we also only do this for select markers, not for the whole panel. And these are typically ones where we want to be certain of the gate placement, or if we want to do like one final check for any hidden spreading. And I just want to note with FMOs, they only define the lowest possible bound of a gate, but often in reality, when you're looking at your entire panel and establishing your gating template, often that gate is going to have to be placed higher to improve the specificity of your marker, or because of shifts in the negative that happen with and without the antibody in your panel during the FMO staining. So now I'm going to talk about how we validated our ICS assay for vaccine clinical trials. At the onset of the COVID-19 pandemic, we developed a new 27-color panel for the detection of CD4-Th1 and Th2 cytokines and CD8 T cell responses. We utilized our experience in designing high-parameter panels for HIV vaccine research and using those principles of high panel design that I just went over. The goal of this panel was to maintain the sensitive detection of CD8 T cells and Th1 cytokine responses that we had in previously validated ICS assays. And then for this panel in particular, we really wanted to optimize the detection of the low frequency and low intensity Th2 cytokines. This is very important in the context of COVID-19 due to concerns of Th2 skewing. So to optimize detection of these low frequency cytokines, we place them on the brilliant blue fluors to improve the sensitivity. And then we also combined IL-5 and IL-13 on the same detector. And here I'm just showing some representative flow plots of the primary readouts of the assay. On the left, we have interferon gamma and IL-2 for our CD4 and CD8 T cells. And then in the middle and the right plots, I have our Th2 markers for CD4 T cells, IL-4 in the middle, and IL-5 or IL-13 on the right. So this is the final panel. The markers in bold are the critical markers in our panel and the ones that we chose to validate, and the other markers we can consider as standardized. And we actually have since developed a next generation panel based on this one, where we substituted in markers for circulating T follicular helper cells. And then we also just recently did an additional update to the panel to replace the brilliant blue fluors with the new real blue dyes that are even brighter and have better spillover characteristics compared to the brilliant blue dyes. We have a very methodical approach to assay qualification and validation. First, we perform extensive optimization and standardization, and this is both for our cytometers and our panels. We base our qualifications or validations on these two sets of guidelines that I list below. And this panel was actually validated twice. The first time we validated it in 2020 using PBMC from HIV vaccine recipients. But when we were collaborating with the FDA, we received feedback requiring us to repeat many aspects of the validation using spike-specific responses, because they said that HIV-specific responses were not a suitable surrogate for SARS-CoV-2 specific responses. And this goes back to the objective of a validation of an analytical procedure, which is to demonstrate that it is suitable for its intended purpose. So as I mentioned, we had to adapt the guidance documents for the validation of this assay. There's some challenges to validating a functional flow cytometric assay. It's not bioanalytical. Each sample contains its own negative control. There's no universal negative. There's also no gold standards or calibration or reference standards. We don't have any results that are based on a curve or compared to knowns. And then also for multi-parameter flow cytometry, your panels can be very large, contain 20 plus markers, and it's not possible to validate them all. So you need to select key functional markers that you want to validate, and then you can consider other markers for qualification, and then everything else would be standardized. Other considerations are that it's crucial to, at the onset, to set your preset your acceptance criteria before embarking on the validation assays. You can't do the assays and then decide what your acceptance criteria are so that you pass. These have to be done before you do the assays. And then you also need access to a large number of samples with known responses. For our validation assays, we use peptide pools to cover the spike S1 and S2 subunits, and we use 3 BD flow cytometers that were validated, optimized, and cross-standardized, and on which we performed daily QC prior to use. Our negative control in the assays was DMSO, which is our peptide diluent. We also included a batch control with each assay, and then we determined positivity using our validated positivity method, which is an adjusted Fisher's exact test. We assess these 5 parameters in the validation, and I'm going to go through them one by one in the next slides. So specificity addresses the ability of the assay to return a negative result for a known negative. And actually, in the context of COVID-19, it's not possible to directly assess specificity because of cross-reactivity to endemic beta coronaviruses. But when we did our first validation, we did assess specificity in the context of HIV. This assay requires a large number of samples that are presumed to be negative. In this case, we took pre-pandemic samples. And while we did have some positive results in the assay that were mostly likely true cross-reactive responses, nevertheless, they all fell below our false positive acceptance rate of less than 10%. The precision validation assesses the degree of agreement of results obtained from repeated testing of the same sample. So we accomplish this by doing 6 independent runs of each sample using multiple technicians over multiple days, and each sample was assayed in triplicate. And our samples were pre-selected to have a range of responses that we would expect to see from vaccine-induced responses. And our acceptance criteria for this testing was CVs across all runs of less than or equal to 35% for 80% of the values, which may sound a bit strange, but this is based on our experience with the assay that showed this is how the assay performs. And here I'm showing the precision results for CD4 T cells expressing interferon gamma and or IL-2 in response to total spike on the top, and then spike 1 and spike 2 individually in the middle and the bottom. And the responses are ordered in decreasing magnitude. And we are looking for where the interassay CV becomes greater than 35%. And this is actually was not reached in this set of testing. And we were precise in our assay down to responses of about 0.032%. And so we actually had to assess lower level responses in other assays. The sensitivity validation is comprised of two components. We have LLOQ, the lower limit of quantitation, and LOD, the lower limit of detection. Because of time, I'm only going to show results from lower limit of detection, which is the lowest response that can be consistently detected. So the limit of detection has two definitions. The first one is the analytical definition, which is based on the distribution of the data for the blank. And in this case, in this assay, it's the negative condition for each cytokine of interest. And these are the values listed in the green box. There's not a fixed value. There's a different value for every cytokine and for every sample. We also developed a functional definition of limit of detection, and this is based on the lowest magnitude of response that is determined to be positive by the Fisher's exact test. And these are the values that are in the red box. And these typically are around 0.05%. And this is consistent with what we see in our LLOQ assays and also our precision assays. The linearity validation assesses the range of responses for which the assay yields a reliable result. For this, we serially diluted stimulated PBMC into unstimulated PBMC and tested triplicates at each dilution level. And for the linear fit, we took only points above LLOQ and with an interpoint CV of less than 30%. And here I'm showing the linearity results for CD4 T cells on the top and CD8 T cells on the bottom, for S1 stimulation on the left and S2 stimulation on the right. And the expected responses are on the x-axis and the measured responses are on the y-axis. For both CD4 and CD8 T cells, we maintain linearity down to about 0.01%. And accuracy validation assesses how close positive responses are between a validated assay and the new assay, with the goal of establishing equivalence between the two. And this is established if the 95% lower bound of the concordance correlation coefficient is greater than 0.7. For this validation, we used interferon gamma and or IL-2 responses to HIV peptides and CMV. So we combined the responses from the HIV peptides and CMV and plotted them and combined them in the same plot. And we have the responses from the 17 color assay, which was validated on the x-axis, and our new 27 color assay on the y-axis. And for both CD4 T cells on the left and CD8 T cells on the right, we had excellent concordance, with the 95% lower confidence bounds being 0.91 for both. So at the end of the validation, we were able to validate except for specificity, which couldn't be directly assessed, our ICS assay for LOD, LLOQ, precision, linearity, and accuracy. We gained a lot of key insights from our interactions with the FDA that can be applied to validations in other vaccine realms. In particular, samples used must have responses to the pathogen of interest, and your batch control also has to have vaccine or pathogen-specific responses. Other important considerations that we realized as we were doing these assays is that sample quality is critical. If you have a poor viability in your sample or poor sample quality, this is going to impact the sample's functional responses and thus impact your validation parameters. And we couldn't have done this validation without our standardized and well-maintained cytometers that produced high-quality data. Overall conclusions. Characterization, optimization, and standardization of flow cytometers is critical not only in the clinical testing realm, but really also in the research realm. It's important to realize that brand new instruments are not immune to deficiencies and manufacturing issues, and you really want to assess the performance of your instrument before you put it into use. When you have standardized instruments, the data from these instruments are reliable and reproducible even longitudinally. And if you have multiple instruments and you standardize them to each other, you can use them interchangeably and you're not just married to one instrument and in a bind if that instrument is down. To design a successful high parameter flow cytometry panel, a methodical approach is needed. And I've shown that it is possible to validate a highly complex flow cytometric assay and perform it in a high throughput manner. And T-cell data produced from our validated ICS assay has been used in support of COVID-19 vaccine authorization in both the US and the European Union. And this is a lot of work. So for researchers not considering a full validation, there are selected aspects of the validation that can be implemented to qualify your assay, because not only in the clinical realm, but also the research realm, high quality and consistent and reproducible results are necessary. And I want to say thank you to all the members of the HVTN laboratory, particularly Julie McElrath and Steve DeRosa, all of our collaborators, the statistical support that we received from multiple teams, and of course our funders without whom we couldn't do this work. Yes. Yes.
Sample was below a certain viability.
Yes. So if a sample had a viability of less than 66%, it wasn't plated in our assays. But actually, we have found that is our acceptance criteria for moving forward with a sample. But actually, we do find that even samples with higher viabilities than that can basically like tank or perform poorly in the assay, and that will also affect the outcome, even if your sample passes that acceptance criteria at the beginning.
So in the end with your staining, did you have a viability cutoff? Like if a sample's viability was less than a certain amount, did you then discard the results?
We didn't have that in our assay, no. Or we
Okay.
didn't have that acceptance criteria.
I was just curious. Thank you.
In a similar vein, a lot of like debris and cellular kind of components can cause issues with your data. Do you have any way of assessing that as a kind of similar to your viability?
Well, we don't have particular cutoff for aggregates or anything like that, but I didn't show it, but we do have an extensive cleanup gating procedure that we go through with a lot of gates to basically exclude all the debris and non-specific staining that we see, so that when we're actually analyzing our cellular populations of interest, they're clean. Thank you very much.